The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
Dr. Rosa Krajmanik-Brown is a professor at the School of Sustainable Engineering and the Built Environment and the Sweetie Center for Environmental Biotechnology at Arizona State University. She has a PhD in environmental engineering from Georgia Tech. She was awarded a National Science Foundation Career Award and was selected as one of 40 under 40 leaders in Phoenix, Arizona. Funding for her research has come from many federal agencies, including the National Institutes of Health, the Department of Energy, the Department of Defense, and the National Science Foundation. She pioneers research on the gut microbiome and autism and is the author of 3 patents and more than 90 peer-reviewed publications. Dr. Krajmanik-Brown specializes in molecular microbial ecology for bioremediation, the use of microbial systems for bioenergy production, and the human intestinal microbial ecology and its relationship to obesity, bariatric surgery, and autism. This webinar is made possible thanks to generous donor support, including a grant from the Local 25 Boston Teamsters. If you'd like to contribute, please visit our website at autism.org. And now I will turn this over to Dr. Krajmanik-Brown.
Thank you very much. Thanks for the invite. It is my pleasure to be here. I'm going to talk to you today about, the title is Research on Microbiota Transfer Therapy in Patients with ASD. But I'm going to be more broad than that, and I'm going to give you an overview of research that has happened in my lab on the microbiota and ASD also, before we talk about the microbiota transfer therapy. I want to start by acknowledging a lot of people that have contributed to this research. I want to thank all the families who have participated and have made this research a reality. Our donors, University of Minnesota and Prestovo Finch for giving us the source of material for microbiota transplant. Dr. Jim Adams, who many of you might know, he's right here. He is one of the reasons why I do autism and microbiome research. Dr. Daewoo Kang, who led most of these studies. A long list of collaborators. Sources of funding, we have ZUBOC for autism, Arizona Board of Regents, and ARI. You can see it here on the bottom. ARI was the first foundation, or I can call it maybe agency, that funded the autism research that happens in my lab. So I'm pretty happy to be here and to give something back to the ARI and to the ARI community. I'm going to start by talking about what the microbiome is and why we're interested in this topic. When the human genome was sequenced, after the human genome was sequenced, and we had information about all the genes that we have in our bodies, as part of our bodies, the NIH, the National Institute of Health, launched an initiative called the Human Microbiome. The human microbiome, first of all, leveraged on all the technology that had been developed to get the sequences for the human genome, but also really quickly realized that there's trillions of microbes in our body, and that it's a very interesting topic to understand and initially sequence most of the microbes that live in our body. The first stage of this human microbiome project focused on getting microorganisms from different parts of the body, from your oral cavity, nasal cavity, the gut, the skin, vagina, in healthy cohorts, just to characterize what is the community composition of these microbes, what were the pathways, and how did this related with the phenotype of the host. Later on, the second phase of this human microbiome project focused more on dynamics over time in specific groups of patients. So they picked three groups of patients, which was preterm birth, inflammatory bowel disease, and prediabetes. And these patients have been sampled many times just to see, again, community composition, but focus more on what is the function of this community composition and how does it change with time, and what can we do with this to actually eventually have therapeutic approaches that target the microbiome. There's been significant public interest for the microbiome, and these are just a few very important news outlets that have spoken about the microbiome since 2010. And I have a few more listed here that actually have targeted our research in the autism and gut bacteria, which has captured also a lot of interest from the media. Microbes in the human body are very important. We have, like I mentioned before, trillions. Most of them live in the GI tract, in the gut. You can see here a pie chart that shows the percentage of where do we have microorganisms in our body, and about 30% live in our GI. The microbiota involves bacteria, archaea, viruses, and fungi, but today I'm going to focus mainly on bacteria, which is what we've done most of our studies with. And I want to point to your attention that at the genome level, we have so many genes of microbes. We have about 100 times more genes of microbes in our bodies than our own genes. So the microbiome, the genomes of the microbes in our body, and the genes that we have in our body from microbes are 100 times more than our own genome, which just shows the potential that this has for therapeutic approaches. And I've been showing this slide, I think, for the last 10 years since I started working on the microbiome field. And I used to have what is known, but now I changed it to what was known, because some of the things that we thought we knew about the microbiome have changed a little bit, or the perception has changed a little bit. I kept in green the things that were postulated since 10 years ago, and they remain. In red, I have what my lab is most interested in. And in black, I have things that have been kind of wobbling or changing, but I'll go through that in a second. The microbiome is sterile at birth. I believe this is the case. There was an article that said that it wasn't because they found microorganisms in the placenta. That article has been strongly debated. And the debate is that possibly those microbes that were sequenced from the placenta were actually contamination. It develops during the first year of life. The common knowledge is that it develops and changes as children start introducing solids into their, it develops initially depending on how the child is fed, either with breast, how the child is born vaginally or C-section, and how the child is fed, breastfed, or with formula, and then once we start introducing solids into the diet, that also changes the microbiome. It's unique to each individual, we know that, and that hasn't changed. There's a general stability throughout life, and I have general in quotations because you will see that through a treatment with antibiotics, for example, the microbiome changes. Through a treatment, we change the microbiome, but you will see that eventually it goes back to certain stability after the intervention. It's affected by internal and external factors. We know that some of those external factors can be our diet or the environment. For example, internal factors are our genome and our physiology. It's altered in disease. The microbiomes of obese individuals is different than the microbiome of normal weight individuals. The microbiome of patients that have irritable bowel disease is different. And yes, you will see today the microbiomes of kids in the spectrum also is different. And it's strongly affected by residence time, which is an important finding when we're talking about GI symptoms. The microbiome impacts a variety of health conditions. You can see a long list here. My lab has focused in the last 10 years in metabolism and obesity and autism, which is what I'm going to talk to you about today. You probably have heard the saying, you are what you eat. And I usually tell people you are what you eat, mainly because you are, at the genome level, 1% human and 99% microbes. And whatever you eat will craft your microbiome. So in this slide, you can see that what we eat gets to our bacteria in the gut, and that these bacteria actually produce certain chemicals. In this case, these are short-chain fatty acids that interact with our gut lumen, but they also interact with other parts of our body, like the liver and the brain. And this is an important thing to remember for our presentation today. Are we more human or microbe? My lab focuses on interventions. Can we change the microbiome? Can we change these microbes that live in our gut? Some of the interventions that we're looking at are bariatric surgery, which is an intervention that changes dramatically the gut microbiome. fecal microbiota transplant, and diet. And we look at interventions and how can we use this to improve health, and we also look at biomarkers, which are also important for health outcomes. As many of you know, autism diagnosis in the United States has increased dramatically in the last few years. Just from 2016 to 2018, the CDC published numbers that in 2016 we had 1 in 68 school-age children, and in 2018 we have 1 in 59 school-age children. Some of the factors that affect ASD and its symptoms are genomic, environmental, and we are looking at the microbiome. I do want to mention that the amount of ASD that can be, or the percentage of ASD that can be explained by genomic changes in the human genome is about 30%. The environmental exposures are considered to be much stronger than the genomic. And I also, if you remember on my first slide, I said that the microbiome changes depending on external and internal factors. So when we're considering the microbiome, keep in mind that the microbiome interacts with these 2 other factors also. So the bacteria in our gut interacts with our physiology, and it also interacts with our environment. And so it's a composite of these 2 factors also. I mentioned before that Dr. Jim Adams is one of the reasons, or the strong reason, why I started doing research in microbiome and autism. I had already been doing some work with the microbiome, and he published this paper, and we had a conversation about the fact that he had data that showed that as severity of autism increases, also gastrointestinal symptoms increased in a cohort that he studied in 2011. And so this piece of evidence and a paper that was published in 2000 by the group of Sidney Feingold, that showed that kids with autism that were treated with oral vancomycin, their behavior improved, which you can see in this graph. This is a change in behavior. However, their behavior improved during treatment, but once they stopped taking the vancomycin, even though they had a probiotic after the treatment, their behavior actually regressed after the treatment. So there was this provided some evidence that microorganisms in the gut are possibly involved in some of the symptoms and are possibly sending some messages through the gut or to the brain. As I mentioned before, my group has been studying the gut microbiota, and we know that microorganisms do a lot of things for us. Simply, they help us digest our food, but they also produce vitamins, and they produce chemicals and metabolites that interact with our brain and with our immune system. They also protect our colonocytes, which is another important thing that they do. So the first thing that we did, which was actually funded by ARI, was looking at if there was a difference in the gut microbiome of kids with ASD and neurotypical children. And what you're looking at in these 2 graphs are diversity. Diversity is an index of how many different types of microbes do we have in our intestine. In red, you can see the kids with autism, and in blue, you can see the neurotypical children. And using 2 different indexes, you can see that diversity was higher on the neurotypical or typically developing children than on the kids with ASD in this cohort. We also found that these children that had lower diversity, we started looking at specific microbes, and we saw that there's this microbe called Prevotella, which is a fermenter that really likes fiber, for example, was very highly depleted in children with ASD. In this graph, you can see here on your right, these are typically developing children, and here are our children with ASD. And you can see here the abundance of Prevotella on this plot, and you can see the depletion here on our kids with autism. You can also see in this graph, which is a little bit busier, but again, you have your typically developing children on the right and your kids with autism on the left, that there is like a yin-yang relationship between this group that contains Prevotella and a group that contains a few pathogens, including Shigella and Enterobacteriaceae, which is a group that has a lot of pathogens. And you can see that, for example, in this group where we're missing the beneficial microbes, we have an abundance of the harmful ones. Same here, you can see we have the beneficial one and the harmfuls are missing. Harmful where we're missing the beneficial. So there seems to be a yin and yang correlation where these beneficial microbes seem to be protective against these harmful microbes. We had findings on this small cohort that was 20 children with autism and 20 typically developing children. And we went to a second cohort that had independent 20 children with typically developing and 20 kids with ASD. And we were able to replicate, again, higher diversity for the typically developing than the autism. And when we look at this microbe Prevotella, which was one of the ones that we've been tracking, we also had higher on the kids typically developing than on the kids with autism. And so this finding is very interesting and very exciting, but I want to point out that many other groups have also been looking at the differences between kids with autism and neurotypical kids. In 2015, we published a review where we look at all the other articles that had been published at the time. And we saw that there seems to be controversy between the findings on different groups. And so some of the findings that we have are that we have increase of harmful microbes like Suterella and Clostridium. Other groups find lack of beneficial microbes like we did, like Bifidobacterium, which is typical probiotic, and Prevotella. We have many studies and different findings, and there might be issues confining these differences or that could explain these differences. Some of them are the techniques used, some studies use siblings as controls and some don't, and a few other things that we explain in this paper. But I just want to point your attention that we're not the only ones that were looking at this question. Others have, and the findings sometimes are different. Okay, so we found that kids with autism had lower diversity and they were lacking beneficial microbes. I told you many times that we are 1% human and 99% microbe, and I think that's one of the reasons why your DNA isn't your destiny, because we can't change our DNA, but we can somehow modulate and change the gut microbiome, which is a big part of our DNA. That's why it's not our destiny. So we went into a journey of trying to change the microbiome in kids with autism that had GI, gastrointestinal symptoms. And we published in 2017 a paper, Microbiota Transfer Therapy, alters gut ecosystem and improves gastrointestinal and autism symptoms. And I'm going to show you some of the key findings of this paper. It's I'm mentioning that it's an open label study because that is important. But before I move to the findings of this paper, you might have heard, some of you might have heard that there have been issues lately with FMT. And this is we call it a modified fecal transplant, even though we don't call it directly a fecal transplant. And the media has been really concerned about the fact that a patient died after a fecal transplant. And this happened because this fecal transplant, first of all, was done in a patient that was immunocompromised. And second of all, the material used for this fecal transplant had a microbe that was antibiotic resistant, which led to the death of this patient. And so I want to make sure that when I show you our results, which are really exciting, you understand first that we use very safe microbes that were actually tested for antibiotic resistance and for many other things that were tested for known pathogens, and they were very clean and very regulated and very tested. And this is one of the reasons why I always tell, especially families, do not try this at home. It's very important that this is done with very clean supervised material and under medical supervision. So now I move into our exciting treatment. So we had 18 kids with ASD. They all had GI symptoms. And we also had 20 neurotypical kids, which we did not treat. The neurotypical kids were not treated. They were just used as a standard control of how the microbiota changes throughout the treatment, but they were not treated. And the treatment involved first vancomycin for 2 weeks. Vancomycin was used to get rid of the bacteria that was in the gut. Then the children had Moby prep, which emptied completely the bowel. This is what is used before colonoscopy. And they also had Prilosec, which is a proton pump inhibitor, which helped us provide the microorganisms, this microbiota transfer, because some of them were provided orally. First, the children had an inoculation, or they were provided the microbes with a high dose. Some of them was oral and some of them was rectal. And then after that, we had 7 weeks where the children drank a maintenance dose every day. And so this is one of the reasons why we needed the proton pump inhibitor. We took stool samples, swab samples, and we have a few assessments. I have more detail here on the treatment. So in the assessments, we assessed the GI, and we had certain assessments for behavior, of course. And with the stool samples, we were able to look at the gut microbiome, and we're investigating metabolites. We took important samples in the beginning, after vancomycin, which is or high inoculation, at 10 weeks, which is at the end of treatment. And this graph also shows you that we follow the children also for another 8 weeks. This is important because if you remember on the vancomycin study initially, the kids actually lost the benefits after the treatment. So we wanted to see what happens 8 weeks after the treatment. And we also took samples here for microbiome analysis. And before I show you the results of our microbes, which is what I will tell you first, I want to say that this is the first clinical trial where I have been involved where we had 0 dropouts. That's the first result that I want to tell you was amazing. No one dropped out of the study. 18 children started, 18 children finished the study, and this was intensive. We actually asked a lot from our participants. And so if you remember from our previous studies, one of the things that that we wanted to fix in the kids with autism was microbial diversity, the amount and types of microbes that live in the gut. Here, let me explain what I'm showing you. This is FAITH-PD, which is a measurement of diversity. Higher is better. In this line, this orange line is the average diversity of our high dose donors. This green line is the average diversity of our neurotypical children. And here's the average diversity of our maintenance donor. The children with autism start here, week 0, and you can see that the diversity is lower than the neurotypical children, the typically developing children. The diversity drops a little bit at week 3, which is not surprise. They just had 2 weeks of vancomycin that killed a lot of microbes in their gut. But then after that, as we provide the high dose and the maintenance dose for 7 weeks, their diversity increases dramatically. So you can see that at week 10, which is at the end of the treatment, the diversity is pretty high and close to the kids that are typically developing. And then 8 weeks after treatment, it's even higher, which was really surprising, amazing, and exciting. If we look at how much did the microbiome of these kids that receive donors, healthy donors microbes, resemble the healthy donors? I'm showing in a graph here UniFrac. UniFrac is a measurement of how similar is something, but it's really a dissimilarity measurement. So the higher the number, the less similar things are. The lower the number, the more similar things are. You can see the kids typically developing here, and so you could take this as this number is the natural variability between individuals. If we compare me and you, we're probably going to come up with a number that is similar to this. So here we compare the children with their most recent donor, and you can see that in the beginning, of course, they were very different to their most recent donor, some of them up to this high. After vancomycin, they actually started, and the high dose, they started resembling more their donor. And after the 10 weeks of treatment, you can see that the number is lower, which shows that they resemble the donor more than the natural variation that we have between individuals. So they were more similar to their donor than a person it is similar to another person, a child their age similar to another child. And you can see that this number bounced up a little bit, but it remains pretty similar. So it does look like maybe it's going a little bit up after 8 weeks. We also look at specific microbes, Bifidobacterium, which is a beneficial microbes. Here is the relative abundance, initial numbers on our kids with ASD. After treatment, you can see how it came up. This is the average relative abundance in neurotypical kids and in our donors. Prevotella, which is one of the microbes that I told you about before initially, and look at the huge increase. This is orders of magnitude after treatment, or even higher than our typically developing kids after treatment and higher than our donor. And the sulfovibrio, which is a microbe that is controversial, but we're also following, it was higher after treatment and higher than our neurotypical and donor. This is surprising, and you can see probably here, well, it's even higher than the donor. What happened? I have it on my title. I think that the gut environment changed with our treatment, and it changed in a way that these children were actually able to invite beneficial microbes and grow beneficial microbes in their gut. And that's why these microbes were happily growing, probably even at higher numbers than what we had in the donors. All right, so I showed you the bacteria, which is what I know the most about and what I like, but you probably want to hear, okay, the bacteria change and the community change, but can that improve gastrointestinal and autism symptoms? And it did, of course it did. So we have here a gastrointestinal symptom rating scale assessment. Our children started at about 3, and a normal GI is at a number of about 1. And you can see that throughout the treatment, this is evaluated every week. Their GI symptoms improved drastically too. And at the same time, we have a parent assessment that was done also every week, which just measures improvement. You can see that the improvement also increases at a similar rate as our GI symptoms are decreasing. We also did assessments that were done by an external evaluator. I think that's very important because this was an open label study where everyone knew that they were receiving the treatment. Everyone was treated, so there could be some placebo effect. We had an external evaluator evaluate the children, and with this external evaluation using CARS, you can see also that there were significant improvements in behavior. This is our baseline at the end of treatment, and amazingly, surprisingly, and exciting enough, 8 weeks after treatment, the improvements not only remain, but seem to be going even farther. And so not only the fact that these improvements actually seem to be getting even better 8 weeks later, but after we finished this treatment, we had families that were calling Dr. Adams, who handled all the recruitment and communicated with the patients, telling him that the kids were improving even more and more and more. So he said, Rosie, we need to do a follow-up. We need to come back to these families and we need to see how are they doing. How are they doing? Initially, we wanted to do this a year later, but by the time we got permits, et cetera, this was a 2-year follow-up. And we called back, or we got approval, which you need to get approval first to call the families. We got approval to go back to our families and ask them if they would be willing to provide samples again and fill in questionnaires and do assessments again for their children. And amazingly enough, all 18 families came back to participate, which was surprising and incredible. All 18 families participated for all the behavior and GI assessments, but for the microbiota, we received samples only for 16 families, but it's still pretty good. 2 years later. And so the results on our diversity, you remember that this tells us how diverse our samples are, and the higher the better. We started here, and after 8 weeks after treatment, the diversity was higher than when the treatment ended, and 2 years later, it was even higher than when we finished the treatment and 8 weeks after treatment, which is amazing and it's exciting. And this also, I think, goes to show that the gut environment change. And this is something very important, because if you change the microbiota, but the environment is still not conducive to good microbes, then this would revert to have a bad microbiota again. And you can see that the diversity here is even higher than the diversity of the typically developing children, which are on the green line here. This is our collaborator. I need to mention him, Greg Caporaso, who helped with a lot of the analysis for this part of the study. And another thing that happened that was interesting is that at this time, the recipients of the microbiome shifted away from the donor. So if you remember, this number of unifrac tells us how similar they were to the donor, and lower is more similar to the donor, which is what we see after treatment. But after 2 years, they actually, how different or similar they are to the donor is the same as the difference that we see, the average neurotypical difference, or the average difference that we would see between you and me. And so these children established a new steady state that was a new beneficial, healthy steady state that was their own. It didn't look like the donor anymore, but it was a good, healthy steady state for themselves. And when we look at these beneficial microbes again, here we have week 0 or week 10 and 18 for Bifidobacterium, and 2 years later, Bifidobacterium actually numbers were even higher than initially. This happened mainly for Bifidobacterium. When we look at Prevotella, the numbers of Prevotella are still higher than at week 0, but our Prevotella numbers are not higher than at the end of treatment. So you can see that they dropped a little bit, but if we compare week 0 to here, we're still much higher, and we're still higher than the neurotypical kids. So that's a big improvement. And or the sulfovibrio also dropped a little bit, but it's still also higher than week 0. And what happened with GI symptoms? On GI symptoms, the children were regressed a little bit compared to the end of treatment, and we had seen that a little bit 8 weeks after treatment. But this is much better than in the beginning. So our children are not at 1, which would be perfect, but they're much lower and much better than how they were when they began the treatment. So the GI is still much healthier than when they began the treatment. And if you're curious about what are some of the things that change in the GI symptoms during treatment, abdominal pain, you can see that it's still much better even after follow-up. Reflux didn't change a lot because we didn't have high reflux in the beginning. Diarrhea changed dramatically, and constipation also. Behavior, if we look at CARS, its behavior symptoms improved even more. So when we saw that we had huge improvement 8 weeks after treatment, and Dr. Adams said we should go back and see how our kids are doing later, he was completely right. Our kids were doing so much better that if you look at this graph, I have 2 lines here that cut between severe autism, mild to moderate, and minimal to no diagnosis at all. We have a few children that actually can be considered not on the spectrum anymore after finishing this treatment. These were about 8 children that actually were below cutoff. In a more simplified way, you can see here that in the beginning of our treatment, most of our patients had severe symptoms based on CARS. 80% of them have severe symptoms, and only a few of them were mild to moderate. And when we finished the treatment and 2 years later, a big percentage had minimal to no symptoms, over 40%. A little bit under 40% was mild to moderate, and only a few of them were still with severe symptoms. And this and the previous graph is one of the things that actually has captured a lot of media and families' attention because this seems really promising. Some of the behaviors that we saw changing: irritability, lethargy, hyperactivity, speech, cognition, communication, motivation, and their manners. So this is all really, really, really exciting. But again, I want to mention that this was an open label study. So some of these results need to be taken with a grain of salt because there was no placebo arm, which is how a clinical trial needs to be done on the next stage. And we want to make sure that do not try this at home because this needs to be done with very clean microbiota, which is what we did, very well screened microbiota and under medical supervision. And what needs to happen next with this transplant therapy, MTT, is we need to have a clinical trial where we have a placebo arm. So we are right now working on adults clinical trial with a placebo arm, and we're fundraising for the child clinical trial with a placebo arm, which is the next step that would take to prove the scientific validity of our results, but also to take us closer to taking this to patients, to be approved by FDA, to be able to take it by patients. My lab is very interested in not only can we treat and can we change the gut, but how is this happening? We're very interested in the mechanisms. We're interested first in which are the microbes that are changing, which I already show a few that we're following. But we're interested in saying, how can this be happening? How can the microbes in our gut be perhaps communicating with the brain? And there are a few hypotheses of how microorganisms that live in our gut and our intestine actually communicate with the brain. Some of them are through the immune system. Some of them are through the enteroendocrine cells in the gut, through the vagus nerve. But my lab is looking at metabolites. What are chemicals that these microorganisms are producing that perhaps can be interacting with the brain? And there are some chemicals that we already know that interact with the brain, like gamma aminobutyric acid, fatty acids, 5-HT precursors, which lead to production of serotonin. You probably have heard about serotonin. So we're looking at all these chemicals and trying to figure out, can we get to something simpler on how these microorganisms interact with the brain? And just to give you a snapshot, we also look at metabolites in this treatment, and we look at plasma metabolites and fecal metabolites. And we had 620 metabolites identified in the plasma and 669 in fecal samples. But I'm just going to point your attention to some metabolites that were that changed, or show you that things changed through treatment in the blood mainly. You can see here, this is heat map that shows in pink metabolites that were at higher concentrations, and in blue metabolites that were at lower concentrations. You can see here a spot in our children with ASD where we have higher concentrations at baseline. And you can see that after treatment, that these concentrations or relative abundances came down and are more similar to our typically developing children. And there's also a group of metabolites that were actually lower when compared to neurotypical children in the beginning, and after treatment, they increased in relative abundance. When you look at the metabolites in our fecal samples, there's no clear signature like you saw on the blood. So here we need a microscope probably, or something else, and specific hypotheses to really find if there's something here that can lead us to a solution to treat ASD. I have this slide here just for perspective. I wrote a commentary in the beginning of this year for a paper that was published on behaviors with flies. And this is a fruit fly. And I just want to show you this as a perspective of how far ideally we would like to get if we can, though humans are more complicated than flies. This is a fruit fly that when the fruit fly has its normal microbiome, the fruit fly has a normal behavior. If you have a fruit fly that doesn't have microbes. This is a fruit fly without microbiome, without any microbes. This is a sterile fruit fly. This is a hyperactive fruit fly. Okay, so if you take this hyperactive fruit fly and you add one microbe that was here, which is actually a lactobacillus, Lactobacillus brevis, just this lactobacillus, the fruit fly behavior becomes normal. So it only takes this one microbe to actually modulate the behavior, and it loses this hyperactivity. If you take this fly actually that has normal behavior and you add an antibiotic, it also becomes hyperactive. So this is a key finding that, again, the antibiotic is changing the behavior, as we saw with the vancomycin, and this tells us that microbes are involved. And this paper actually took it farther, and they were able to identify the specific microbe, which is lactobacillus, and they were able to identify an enzyme, a protein that actually changes sugar. And this is what causes this hyperactive behavior. The sugar is trehalose. So it would be great if we could find the specific metabolites and the specific enzymes and the specific microbes that can do this. And the complexity of human bodies, because of that, we probably have more than one microbe, which is what I think. So this is just for perspective. So with this, I just want to have a quick summary of our findings. Abnormal gut bacteria in children with autism, including lack of beneficial bacteria. This is one of the things that we found. MTT is a promising method to restore these normal bacteria or microbiome. This improved gut symptoms and possibly autism symptoms, because as you improve the health of the gut, this is what happens. We need more research. We need larger placebo-controlled studies to verify these results and to hopefully get to mechanisms that show us exactly which microbes and which chemicals are involved in these pathways. That's what we're doing. We're looking for mechanisms. And with this, I would like to conclude my presentation and take any questions.
All right, great. Well, thank you for that great presentation. There was a lot of information. We've got a lot of questions. I'll just launch right into them. The first one is about donor matching. So they're asking about whether the recipient and the donors have to be matched and how that screening process works. Also, how do you screen fecal matter to make sure that it is safe? What's the process that you actually use to do that?
Okay, so first of all, they were not matched. There was no matching. So we received the samples and we used them. There was no matching at all, which is something that could be potentially done in the future through personalized medicine, but there was no matching. And there was no matching, and our samples came from adults, which before we started the treatment, this was criticized. We were criticized by that because we were using adults' microbiota to put it into children microbiota, but it worked. Okay, how do we screen it? We get the material already screened from University of Minnesota, but my understanding is that they actually send they clean the product, they clean it in a way that the product doesn't have any solids and it's basically 99% microbes. After they have that product that it's 99% microbes, they use commercial labs to look for viruses, pathogens, and I know that they also looked for antibiotic resistance. I didn't mention on my presentation, but since you asked about that, I'm going to add that the FDA actually put a halt in any clinical trial that was going on with FMT after they had this patient died. We got a letter from the FDA for our adult trial saying you can't do anything right now until you answer these specific questions. And we were able to answer and reply to the request really easily, and the halt has been lifted for us because our product was so clean and so well screened.
The next question is about diets. So the people who were enrolled in the study, the children, were they documented for any special diets that they were on, or is that data that you just excluded from the study? I know a lot of families use gluten-free, dairy-free, different types of diets. So was that included in the design, or was that just not part of it?
So what we did for the treatment, we every family did a diet diary. And so we took diet diaries to analyze what their diet was about. In our first study that was funded by ARI, when we were just comparing the different microbes, we only had something simple as, are you in a lactose-free, dairy-free diet? Are you eating less fish? It was very simple, but we realized that in order to connect better the microbiota, which diet is very important for the microbiota, we wanted a better assessment of their diet. So they actually filled in diet diaries. One handicap of our study is that we did this in the beginning and we didn't do it later. And so I wish we had done it later, because I could hypothesize that their diet changed, and that this was also one of the things that led to this beneficial environment and beneficial microbiota. I could hypothesize that their diet was less restrictive, that once the children, and this is an hypothesis because we don't have the data, once the children were more comfortable and their GI, their tummy wasn't hurting and they were not suffering so much from this diarrhea and constipation and a few other things, and their behavior improved, they became probably more adventurous, trying new foods, and they probably had a more diverse diet, which of course helps the microbiome too. But this is an hypothesis. We didn't check diets at the end of treatment, which in hindsight we should have.
The next question is about participants who did not improve. So were there any theories or similarities in the minority participants who didn't show behavioral improvement?
Okay, so we actually, during the treatment, we called two of our participants non-responders, which if I go back to the slides, you will see 2 grade points, but those were based on GI, not on behavior. If you see these slides here, hold on. The gray dots, these gray dots over here, are two patients that we consider them non-responders because their GI didn't improve more than 50%, which everyone else did. So some of that is linked. And you can see that not everyone improved the same way. We didn't overanalyze the data because we only had 18 patients. And so we could separate it and say, well, the ones that didn't improve had this and that, and the ones that didn't improve ate this and that. But it is overanalyzing the data when we only have 18 patients. So this is one of the reasons why we need to run larger clinical trials. So there's little we can say and really support it with statistics when we've had only 2 or 3 people that didn't respond to the treatment and our sample size was 18.
Okay. People are asking about if they wanted to get this type of therapy. Obviously, there has been news about, the FDA put out a warning about it last spring. So I mean, you gave some guidelines, definitely twice you mentioned about making sure that you're working with people who know what they're doing, who are using screen samples. Where are people receiving treatment, or is it on hold right now?
This is not FDA approved. So the FDA the FDA took a stand on FMTs. This was a few years ago, actually, before we even started our study, where FMTs right now are not approved as a treatment. They're only approved as an investigational new drug. So FMTs right now can only be used for clinical trials. And so the reasons we're doing these clinical trials is to prove that it's safe and to prove that there's improvements so we can actually make it available to the public. So FMT is not available to the public. It is not available. That's one of the things that we're trying to do also through these trials.
Right. And as you said, it's not something that we should just try at home.
No,
right.
Definitely not.
So in terms of clinical trials, I'm sure you can guess my next question. Lots of people have asked, how could they find out exactly where trials are happening and how to enroll?
So if they go to my website, which is here on the bottom of the screen, there is a tab on the left that says Autism and Microbiome. And actually in there's a link that tells them how to sign in to at least 2 trials. And actually there's a link for the donations there also. If they go to my website, which is on the bottom, and there's a tab that says Autism and Microbiome. That would be, I guess, the easiest way to reach both the donation and information about future trials.
Okay.
That actually is easier.
Okay, so people had questions about the connection between the gut and GABA and gabapentin. Could you talk a little bit about that, just a little bit more, and how that relates to this study that you conducted?
So we actually looked at metabolites also in our very first cohort, that ARI supported. And we found some differences in GABA, but they were not statistically significant. So GABA is an excitatory molecule, so it has been hypothesized that this could be one of the effectors also. We're looking at other molecules that are also a little bit more complex, which I didn't mention. We might find that it's a molecule that we haven't looked at before. So in GABA also, we didn't find significant differences before and after treatment. Just so you know, we did look into it. But it's not only the GABA. There are fluxes of metabolites because microorganisms produce metabolites and consume metabolites. So we need to understand better how these fluxes work so we know what's happening, and we can maybe modulate the microbiota for this. So like I said, GABA, it's an excitatory molecule that affects the brain. Glutamate, which is part of the pathway also, it's another excitatory molecule that affects the brain. Serotonin is another molecule that affects the brain. There's a few others. And we always look first at the known ones, but we're doing analysis in a way that we can find other chemicals also, even if they have not been selected before as neurotransmitters or neuroactivators.
We've gotten several questions from people about people who are not symptomatic. So if somebody doesn't have any outward GI symptoms, would there still be use in putting them in a trial like this, potentially?
That's another question we want to answer. We would have to do another trial where we have children with and without GI, which would be also a next step, because I hypothesize that it would work also, but we would have to test it. It's a very important question, because this is are the children just behaving better because they feel more comfortable, or is there really a change in chemistry that it's also affecting their brain directly? I would suggest that based on our follow-up studies, we can see that there are behavioral changes that are not linked to GI, because we saw a little bit of a regression on the GI, but we still saw more improvement on the behavior. But we don't have the evidence. So as a scientist, I will say we have to test it.
Okay, what about teenagers? You may have already talked about this a little bit, but what about that age group? I know there's work you'd mentioned about adults and kids.
Well, teenagers were involved in our trial because our trial was, it went all the way to 17-year-olds.
It
so
did, okay.
We had teenagers in our trial.
Okay. All right, so you did tackle teenagers with that one. All right, so this is a question about when you were doing the trial, were you using a liquid or was it a capsule for the administration?
That's a very good question. So now there's capsules available, and for the trial that we're doing right now, we're doing capsules. But when we did the trial, it was a frozen liquid.
Okay. What about enzymes? This person's asking, do you have any information available about people who are in these trials and they're using digestive enzymes or other types of things, or was that controlled?
I don't have information on it, so sorry, can't comment.
Okay, and same with probiotics. I received several questions about that as well.
So probiotics, for example, on that vancomycin treatment, they use probiotics at the end, and it didn't seem like they work as well. When we designed this study, initially we were going to use a probiotic. That was the idea, because that's the easy path. But I have a strong feeling about probiotics that are found that you can find off the shelf. They're like chicken soup. They can't harm you, but I am not sure exactly what they do. And if you think about it from an environmental perspective, which is what I usually do, the microbes that are in probiotics that you can buy in Whole Foods or in any store are microbes that come from milk. Their natural environment is not the human gut, it's milk. the microbes that we're using here, their natural environment is the gut, the human gut. So it's a more natural transition for them to colonize and to thrive and to be happy when they come from their natural environment. So probiotics could help. I'm not saying they can't, but I don't see the potential of them having the impact that we saw here because of those reasons.
Okay. Next question's about, this is a question I got through an email. They're asking about dosages rectally versus orally. Were you doing, can you talk a little about any differences between those two? And then they're just asking about the adult trial. Is it already filled?
I think we have a waiting list for the adult trial.
Okay.
We're not done with it. We have a waiting list. So yeah, so I asked that first because that's an easier question. For the other question was orally or rectally. We didn't see differences, but our sample size is small again. So we tried both. We didn't see differences. Sample size is small. For the adult trial and for the future, we're using capsules, which makes it much easier. It also makes it easier to have a placebo arm. Because when you have to have an arm that, a group in your clinical trial that, you have two groups, one that is receiving the treatment and one that is not, but they don't know who is whom. And so if you have it in a capsule, it's easier to give a capsule that just doesn't have the medicine, the drug, the microbes. So that's the easiest way to do a placebo-controlled trial with just capsules. So for the future, we're not doing rectally, we're just doing capsules.
So just time for a couple more. The first one here, they're asking about yeast. Was there any discussion about yeast in the gut before or after? Was that excluded?
There was no discussion. We actually have preliminary data on our previous cohorts where we see more candida in the kids with autism, but we didn't look at it yet on these samples. It's a very good question. It is a very good and valid question. And that's one of the reasons why I also mentioned that I was going to focus on bacteria in my presentation, because yeast is fungi and it is important, but we haven't tackled that question yet. We do plan to do it.
All right, you described fruit flies that were sterile, that had basically zero microbes. So were there any kids who, obviously they didn't have zero microbes, but were there any that had zero of the healthy microbes? So like Bifido or any of the other at the beginning?
I don't think anyone has zero, just have lower amounts. But we with Prevotella, we saw some that have none.